Okay, welcome. Welcome. And my name is Dave Champlin. This is BIO409 and 509, cell molecular biology. If you're in the lab class, we're not going to meet this afternoon. Our first meeting is going to be Thursday afternoon. And I posted a Brightspace announcement about that and what you need to bring to class on Thursday afternoon if you're in the lab class. It's an option. Not everybody in here is in the lab class. Okay? And USM had some really excellent clubs before the pandemic. The pandemic, of course, crushed those everywhere. And it's hard to get them going again because during the pandemic, students and everyone got used to not, providing any kind of time in their schedule for just kind of socializing. But it turns out student clubs are great. So some students are kickstarting the pre-med club. And there's and I have some of these. There's some signs up too in the science building. If you want to scan it, you can just come by, or you can just take them because I have a few. Okay, so that's that. And here we're just going to start out talking about what you should be doing this week. Okay, so I have a list of things for you to be working on this week. The first is to get the textbook, and we can talk about it. I switched to the most recent edition of the textbook, the 9th edition. It's been out for a couple of years, so hopefully you're able to find some used copies. And I posted an announcement on Brightspace about the details and explanation for why we need to have it. So I don't use Brightspace very much. I don't use it for grades, for example. There's a long story about why I use it for this and why I don't use it for that. But one thing I do is I post numbered announcements on Brightspace. So there are already some announcement up there, and you should look at them. Okay, and so here's just an example of what the list of announcements looks like. So I've included some information. We're going to have some quizzes next Tuesday, a week from today, and next Thursday. And they're based just on these videos. So if you don't happen to have the book quite yet, you can get ready for the quizzes. I consider them to be prerequisite knowledge, right? This course has some prerequisites and nothing fancy, just get ready for that. They're going to be short answer quizzes. The syllabus is there. I posted the syllabus, and I think I have just a little bit of information about the book here, but it's all on the line. And then I'm not handing out a syllabus because I don't print stuff out anymore. I just post it on Brightspace. And so there it is in our course and under content, and there's the syllabus. And the syllabus starts with a video I made talking about the syllabus, and it's just 10 minutes long, and it explains the details of what you're expected to do for this course. So I won't talk about it in class. I can certainly answer questions in class, because if you ever have a question, you should bring it to class. You're welcome to email me the question, but it's great to bring it to class also because other students will wonder the same thing. And I won't have noticed it, But so there's a little YouTube video. These are all just YouTube videos of just talking through the syllabus. And we'll get rolling on that today. So then the third thing you should do this week is watch these videos. I think they're a total of, like about half an hour of videos, short 10-minute videos for Tuesday and then for Thursday. And the quizzes will be at the end of class, so you'll have plenty of time to watch them and come prepared to class and ask questions in class, like that. And again, if you have questions when you're watching any of this, write it down and bring it to class. Again, you're welcome to email me. I should mention I'm currently the chair of the biology department, and I have hundreds of emails. So I'm a little bit slow to respond to email. If you send me an email and I don't respond within a day or two, just send the exact same email. Just send it again because it's been scrolled way off and I'm scrambling to put out fires. So that's just it. And so I'm busy with that, but I also have lots of time to meet and talk with you. And so we'll talk about that as well as we get into things. Maybe I'll just mention it now. So I have some office hours I post, but I have so much stuff. I don't sit down and wait during office hours. I'm just always on the move. However, I almost have nothing scheduled up until a couple of days before it's happening. So we can meet, and there's tons of time we can meet, and we can talk about anything. We can talk about this course, we can talk about your other courses, We can talk about career path options. I've been a professor here for a long time, and the opportunities for STEM students at USM are just out of control, fantastic. And so I have a lot of experience talking about that and giving some hands-on experience while you're in school. There are just lots and lots of opportunities. USM is almost unique among all colleges and universities in that USM pays students to do off-campus internships. You get paid to do stuff on campus too through the Europe program, but you're paid to do stuff off campus. It's just crazy, and it's because we're in a place that's populated, right? I mean, there's no other place in northern New England that's anything like this, where they have access to these opportunities off campus. Lots of times they're paid by USM or the host. Sometimes they're volunteer because they're so worthwhile. So I have lots to say about that. So we could talk about that, right? And that's pretty clunky to talk about an email, but we could schedule a time to meet in person. So just send me an email, suggest a couple times you could meet, and we'll make a time. We can meet in person or via Zoom or phone, whatever you want to do. Yep. So the next one is read chapter 1 and take notes on it. So anytime you're reading science, you should be taking notes. It's a joke to think that you could read science, or anyone, I can't read science without taking notes, to keep engaged in it. And in taking notes, of course, write down questions. There'll be questions on our exam that we just didn't have a chance to talk about in class. And they'll be from our book. So you really need to get the book and you need to study it, learn about it. However, like all the biology, the textbook has more information than we could cover in class. So it's challenging to tackle the book. The highest priority is the stuff that's happening in class. I always tell my classes that if a student understood what happens in class, I think they should get a final grade of a B, like that. That's just great. To do that, you have to look at the book also. You can't just come to class and hope that you're all set. So you got to look at the book one way or the other. One, to confirm and further understand what we talked about in class. Two, to help kind of cover some of the stuff that we didn't cover in class. Now, there's way too much information for me to ask trick questions or, detailed questions, right? There'll be specific questions, but there'll be some of the more important concepts that we didn't have to cover in class, and we'll talk about that a lot. I remember when I was in college taking classes and how I felt as a student, right? And I'm responsive and sensitive to that. So read chapter 1, and as you're reading it, and write down these questions. Read in the class like that. And if you don't have the book quite yet, I was able to have the library put the 1st and the 7th chapter on electronic reserve on our Brightspace page. So you can read chapter 1 this week. That's what that looks like. So under content, where you find the syllabus, there's electronic reserves, and there's the library page that's got the 2 chapters. I can't put more on. I can't just, I put the checks on, you don't have to buy it. We just rip them off the publisher, right? I can't do more of that. But the first 2. So we're going to read the 1st chapter and then the 7th chapter. The 7th chapter is about the topics that will be for our 2nd quiz next week. And so those 2nd quiz next week is both a kind of background prerequisite knowledge, but it also steps it up to the level we're going to be starting in our course. And that's where the 7th chapter is. So we go chapter 1, chapter 7. And this is actually the order of chapters we're going to read to begin our course. So 1, 7, 16, 17, 18, 19. One thing is that a couple of students have already asked me, the 8th edition is way cheaper. Could I get the 8th edition? Well, it's okay. The changes were mostly in other chapters than the ones we're going to cover. We won't be able to cover everything, and the other chapters were mostly in those. There are some changes in the chapters we'll cover. But you could, conceivably use the 8th edition if you, absolutely have to save more money on the book. Here's the thing, though, is then the other chapters, they've merged some of them, and so the chapter numbers are slightly different. So the topic titles for the chapters are the same, but they'll be off by 1 or 2. So I'll always just be referring to chapter 16, and if you have the 8th edition, you'll just have to be ready to be like, oh, I know what that chapter is. Yep. And again, don't use the 7th edition. So that's that. Read chapter 1. And then here's the next thing. So I record these class meetings, and then I immediately post them on YouTube. And there's a lot to say about it. There's some really significant stuff to say about my expectations of you that's in the syllabus. So what's up with that? So first of all, once in a while that tag fails. I failed to get the recording, and it's just gone, right? So I try my best to get it, and then I post it on YouTube. And I make a Brightspace announcement with the link, like that. So I'll put a Brightspace announcement up, and it's just, here's a link for today's class meeting. It's not an online class. USM really wants students to attend in person, and we'll do stuff in class that will be a reason to be here. Here's an example. Next week, if you miss the quizzes, anytime in class, if you miss an assignment, and everything is announced ahead of time, there's no makeup. I just don't have time to, try to sort out makeup. They're relatively low points when we do stuff in class, and if you just miss class, you miss class, like that. You're required to attend class, and so this is described really carefully in the syllabus. You're not graded to attend, it's part of the course requirements, like that, okay? And if you miss, and it lists a number in the syllabus, and I think if you miss 7 class meetings, well, if you miss more than that, then you could get a lower grade, even if you're doing fine in the course. You gotta try to come. It's understood you're gonna miss some. You're gonna be sick, the car's gonna break down. When that happens, you don't have to tell me ever. I'll have a sign-up sheet and hand that around, and if your name's on it, you're all set. If you miss, then what you need to do, and again, you're not graded on it, you're just required to do it, is to take notes on the recording within a week of the class meeting and email me pics of the recording just to demonstrate that you did participate. And the reason for that is, again, I said, some of this stuff depends on the book, but the most important stuff by far is our class. It would be unfair for me to not provide this resource, because when you get notes from another student, it's just not the same. If you miss class and you get notes from another student or 2 students, you've missed a bunch, right? So we gotta do it. The tech is there, we gotta use it. Still an in-person course, but we gotta use the tech. There's a bunch of reasons why I use it. Here's my favorite one. I'm supposed to be trying to teach students that might become scientists. And if a person is training a scientist, they want these 2 features most importantly. They want someone who is skeptical. So anytime I say something, you just want to start Googling and challenging it and saying, like, that's not what another professor taught, right? That's a good thing. And when you do that, you're not paying attention to class. You're really distracted, right? Another thing is creativity. I could say something, you're like, wow, if that is true, that implies this would be true, and I could potentially start a little company and make some money doing that, right? Those are golden things for a science student. And of course, in other cases, you're just really tired from whatever, right? Yeah. So it's really important to provide that. A lot of times, students will watch this after class anyway, and it might not be all of it. They'll be like, I don't know what was going on that last half of class or that one concept that we were talking about. And within a week, you go back and watch it. I'll delete them after that. So it's these ongoing learning and ongoing participation. And if you don't send me notes, well then that's another reason why it's described on the syllabus that you might not get as good a grade. Now, some people will run into a jam. They'll get sick a bunch, or they'll have some, external issue that's a bunch of stuff. And don't freak out, we'll communicate and we'll work out a plan like that, yep? So that's the next thing is you might just want to watch class again, yep? And I try to talk a bunch about learning and how to learn, because there's a lot of learning that's done in this course, and it's learnable learning, and all of it is very learnable learning, and but it's challenging, so it's a good class to try to learn about it. And one of those things is that, where you just watch it again. So here's the next thing to say. One of these is our book. There's the 8th edition. But there are some different books that are used to teach this course. And I think the next slide is this. I teach this course with a subtitle, and I'll explain to you why it has this informal subtitle today. So it takes a while to lay it out. But this course could be called The Molecular Mechanisms of Cancer. Some students are interested in healthcare careers, so they'll be like, yay, this will be great. Other students are not interested in healthcare career at all. They're interested in plants, you know, or wildlife or ecology like that. That's fine, because if we just chase after this as a common example, it's going to be true for all features of life on Earth. These are universal principles down at that molecular level, down at that nuts and bolts level. So we'll all be fine, but that's what we're going to use as a thread moving forward. And again, I'll describe that. Here's our author, and our author's life story is a story of this studying the molecular mechanism of cancer. And it's really cool, and I don't really care about this person's name or their experiences, really. It's just that what they did is really satisfying when you're trying to explain it to a student. Because back in the 1970s, they finished grad school and they went to Wisconsin to do some research with a famous scientist. And they did a kind of work there that we'll talk about because it's still relevant. And then they moved to Boston and spent the rest of their career there. And when they switched from Wisconsin to Boston, there was a change in their strategy, and I've found really hard for students to sort of absorb it. It's not unique to this person. There was a first amount of research in the 1970s that focused on a certain kind of cancer research, and then this scientist and everybody else shifted, and he made this move. When he was about your age, he made this move, and you know, it's just satisfying to learn that, right? And again, learning about learning, the more we can kind of map it out and make it physical, the more memorable it is, okay? So interestingly also, this guy, and it's relevant for us here in Maine, this guy retired 2 years ago. He was a professor at Harvard and then Boston University. And he moved to Ogunquit, Maine, where he lives, and so, you know, some of you may have mowed his lawn, you know? And he writes now fiction books, and they're thrillers, and they're medical thrillers. And yeah, I think he's a really cool character. Yep. All right. So here's how I get to this explanation of why would we have a subtitle for this course. So we have to get there by this way. And if you've had classes with me before, you've seen this, because I describe biology, the entire study of life, to be a three-legged table. There are just three topics in all of biology, and they fit in this table, and they're equal length. Yeah? Okay? And it's important to emphasize that I just made this up, but it's true. It's just the way it is. And if you asked anyone else, including another biologist, they wouldn't say the same three legs, almost for sure. And they're just wrong, you know, like that. And you're like, ooh, wow. And it's great because it's the first day, right? And I'm driving this car, right? And here are the three legs. The first leg is physiology, and physiology is what it means to be alive. When a cell or an organism is alive, it has this chemical, cellular, tissue, organismal physiology, and when it dies, the physiology is done. So that means, what does it mean when you say, what is life? Well, you talk about physiology. And in this leg, I also put anatomy, so that's the structure of tissues in multicellular plants and animals, and also morphology, which is the overall shape of an organism. And that's a huge, huge leg. And everybody would agree, oh, yeah, that's an old school leg. Most of medicine involves stories of physiology. How's your heart? How's your brain functioning right now? How's it going? Like that, right? So now, and this is the surprise leg probably, is that the next leg, the second leg, is called development. And up until 50 years ago, it was called embryology. So a development leg is about where did you come from? And I teach that in the fall, and you'll see some of the examples we use in this course are from that leg. So I try to avoid talking about cancer in the fall because cancer is about developmental biology, about what goes wrong in development. And I try to avoid that and talk about other topics in the fall, so this is a different course. You'll see that there's some overlap with all these legs in this course, and I'll explain why. The name changed 50 years ago for some really interesting reasons. One is that everybody wondered about embryology forever, right? Aristotle wrote about it. You know, where did you come from, right? Like when you get a haircut and you stand up and your friend says, hey, what's that on the floor? And you're like, well, that's my hair. And they're like, where did it come from? Where did that hair come from, right? And you could use different words. You could say, well, I made it, I grew it, I synthesized it, right? It came from simple nutrients and you put them together, right? So the best verb is to say that I developed that hair, because it's a different thing than to grow or synthesize or any other bird. And that's one reason it shifted, is because it's a different thing. It wasn't here. You could point to your heart and it's the same thing, that you developed that heart. You developed everything else at the same time in your mom's tummy. We all did, but it's a really different thing. And up until 50 years ago, people did not have a clue. Up until 200 years ago, they thought life just occurred by spontaneous generation. 300 years ago, the first person started writing about the idea that fossil seashells on mountains had to have an explanation. And that was the birth of geology. The idea that those seashell fossils were in layers, were in strata, were in sedimentary layers that were stacked based on really long periods of history. At that time, everybody believed in spontaneous generation. So at that time, actually no one was surprised that there were fossils in rocks on mountains, because at that time, everybody thought the earth, like the bowels of the earth, had a generative capacity to make life. And it was just natural for rocks to have what looked like living things in them, right? So, and people just thought that that was like that. The first people that looked in microscopes like 200 years ago, well, yeah, less than that, 150 years ago, the first people that looked in microscopes saw a sperm and they drew pictures of the little guy driving it. But they also believed that. They believed that life came either from spontaneous generation or there was already a little character that just ate and got bigger. Yeah, totally different. The field of development is just dramatically different than it was 50 years ago and up till then. And what caused that is a combination of biochemistry and genetics, like that, coming together with modern tools to tackle it. And these two legs describe everything that ever happens inside a cell or organism on our planet. So it's a lot. It's surprising that those two legs are the same size. And there are explanations for why they need to be judged that way that we could talk about. The third leg is the organism in its environment, like that. And so ecology, and that fits it all together. And one thing that people would say is, if you ask them, what are the three legs? Like a lot of people would say, one of the legs must be evolution. So the deal is, and I have some slides just detailing that we'll loop back to, the deal is evolution is the table. So the table is a real physical thing. It's all the examples of life on Earth, and they all are evolution. And that's because evolution is both a process and a pattern. The process of evolution is what you're used to thinking about. Natural selection, survival of the fittest, is a process that occurs over time. Evolution is also a pattern. It's the product of the process. And everything is an example of that. So this is an important example because you can see the table. It's a three-legged table, and it's evolution. And you'll notice that this table leg was weak. And so up until 50 years ago, the story of evolution was surprisingly wobbly. And we'll talk about that a little bit during our course. But our course really rolls ahead at the nuts and bolts level of life. It's all part of the pattern, but it fits into this big picture. we see these relationships, right? There are relationships between all the organisms on Earth, right? We can focus on humans in our course, but it's telling the story of all life on Earth because it fits this pattern, and the pattern looks like everything is the product of hundreds of millions of years of evolution. Yeah, okay? So let me just loop back to these because I should have shown these little figures. So in the videos for the second quiz next week, I focus on muscles and muscle formation, muscle differentiation. And that's a story in physiology, right? How's your heart, how's your brain, how are your muscles doing? How do they work and how are they working? So a combination of physiology and anatomy, huge exciting leg. If you think about it, you probably have all had classes in college in physiology, maybe multiple classes. It's a huge, huge part. The story development fits in with muscles too. The story in our videos is actually a story of development. It's about muscle differentiation. Muscles weren't here. They had to form and grow and become muscles. And that happens early in life, right? Our heart is not beating as an embryo, and then it starts to beat. That's a muscle. It wasn't here. It starts forming, and then it beats. Then it's true for the rest of our life. As a kid, your muscles get bigger. If you get hurt, your muscles heal. If you work out, your muscles get bigger. Those are all stories of developmental biology, not just embryology, developmental biology. And that's just what the videos are about, is a cell becoming a muscle. And you'll see it's a good example for us because here's a screenshot from one of those videos from our second quiz next week, and it is about the genes that get activated, right? We inherited all the genes from our parents to make muscles. We're just not using them for the first month of pregnancy. And then we have to activate those genes. And molecular biologists are pretty excited about genes, and so I made the videos to introduce that. Yeah, the example is muscles, but the real focus is the genes. And I made the genes out of clay because molecular biologists cut the genes up. And I even start talking about cutting up genes and manipulating them. So cutting them up in a test tube, the DNA is physical, and you can just put them together any way you want. Yep, okay? So here's the deal. Our course is really two courses in one. It's the topic molecular biology and the topic of cell biology. And those and other topics, I mentioned biochemistry and genetics, those are not in the center of biology. These are tools and resources and methods, strategies to study those. Consequently, it's hard to teach those courses because we have to provide examples. There's a really good example about molecular biology in the immune system. Well, a few of you have taken immunology, and most of you haven't. So it's really hard to teach this course, and that's why, the main reason why I say let's focus on cancer, because there isn't another cancer course here at USM. You haven't learned too much about cancer biology. That's a great example, because it turns out molecular biologists, like the author of our text, are the people who just floored it with regard to discovery of cancer biology. 50 years ago, the United States in the early 1970s had great success with getting to the moon in the 1960s and walking around on the moon. The moon mission, the Apollo moon mission, was a great success, and the federal government paid for it, and the federal government said we should turn our attention to another science issue to try to tackle. And so famously in the 1970s, the United States declared war on cancer. And at that time, they provided some funds for doctors, but the bulk of the money at the beginning was going to people that were all molecular biologists, like the author of our book. And they were studying things like in Wisconsin and elsewhere, and they were figuring stuff out really fast to get a foot in the door. Yep. And so it's a great example because it continues to be a story, a lot of molecular biology and cellular biology. Our author of our textbook is a molecular biologist. That's why we go from chapter 1 to chapter 7. Let's do a background chapter, and let's really jump into molecular biology. That's a molecular biology thing. And then we're going to go to chapter 16, 17, 18, 19, which is, our book is organized into 4 sections. And that last section, the 4th section, is about cancer biology. And so the whole way What we're doing is we're foreshadowing that. You know, we're jumping, but you won't have to scramble and read chapter 3 or something like that to pick up some pieces. We have it, we'll have everything we need, but we're just flooring it down that pathway to study the molecular mechanisms of cancer. So that's molecular biology, and I mentioned molecular biology emerged in the 1950s. The story of molecular biology is the discovery of the DNA as the physical primary heritable molecule, and then the study of it as a physical thing. So molecular biology is the marriage of biochemistry and genetics. Those are two really different approaches to biology, and molecular biology is the fusion of it. It's had such a dramatic impact in all areas of biology that almost no one says, I'm a molecular biologist anymore. I don't say I'm a molecular biologist anymore, because it's just so integrated into all of biology. And what it's like is it starts out like we do, thinking about the gene as a piece of clay, and the gene is physical, and it has some different parts that are involved in gene function. And then it says, well, what does the gene do? What's the output? RNA. What's the output of coding gene? Proteins. And so molecular biology just takes off from there and gets to chapter 17 in our book, 16 in our book, really fast. It's about regulating that gene expression story, okay? So then there are topics also that aren't in the center but are related to the center. Ornithology is the study of birds. So you study birds, you talk about those topics with regard to birds. The same is true for the study of fungus. I probably should make a special statement about microbiology, because microbiology doesn't seem to have very much development in it. It's single-celled organisms. But there are the evolutionary precedents for the principles of development even in single-celled organisms. Bacteria talk to each other, and they build little microenvironments that are like a process of development. So microbiology, immunology, I mentioned, neurobiology, other courses like that, especially courses that are really focused on those three topics. And so that's enough of that. It's really said today just to help you understand, oh, it's hard to teach molecular biology because it's off on the side. And we want to talk about genes, and we want to talk about genes in birds and genes in the immune system and genes in the nervous system, you know? But we'll focus especially on genes in cancer just to give us a common thread, kind of that foundational thread. Then there's cell biology. That's the second course that's together here. So molecular biology and cell biology. And cell biology became a field of study in the 1950s when scientists first used electron microscopes to look at eukaryotic cells. And they saw organelles. And they're like, what the heck? There's a lot of organization in a eukaryotic cell. Mitochondria, nucleus, ribosomes. That's cell biology. Cell biology is like molecular biology, where you say, we have to look at the whole cell to figure this stuff out. In our textbook, the 4 sections really have the 3rd section featuring some cell biology. And cell biology is super cool. Students always love cell biology. And we'll cover some chapters. And so what we'll do is we'll do chapter 1, chapter 7, section 4, and then some chapters of section 3, and then we'll be able to pick up some earlier chapters. It's going to be really nice to do them in those ways because we'll have specific examples that we can take with us when we go to talk about cell biology. And when we get to loop back to earlier chapters, we'll have these specific examples we're really solid on that we'll say, oh, now we see we fit it in there. So I've taught this several times, and it works out great. Let me just mention a little bit about myself. So this is a book that a molecular biologist wrote back in the 1970s, a long time ago. And when I was getting ready to go to college, I grew up in Minnesota, a little farming town, and I was outside all the time, whether I was working on farms or just goofing around in the lake and stuff like that. And so when I went to college, my older brother and sister had gone to college. It was just automatic. I was going to college, and I knew what I was going to do. I was going to be a scientist, and I was going to be a scientist because, again, this is a little farming town. Some of you people could relate to this. I had gotten glasses during Christmas break of 2nd grade, and during that break, everyone in my family shrugged and said, I guess you're going to be a scientist because you wear glasses. I was like, okay, well, all right, yeah, you know. And so And everybody else told me that too in my hometown. They're just like, whoa, here comes a scientist, look at you go. You know, like that. Yep, absolutely. And I love nature. So I was going off to college to study the biological basis of animal behavior, like how birds migrate. How does that work? How does a bird know how to do that? That's what I want to do. I want to be a scientist and study that. So I was doing that, and my second year at college, I just happened to pick that book up in the library and read it. And it's a book of molecular biology. And I actually was, like, I read for 10 minutes just standing there. And I said to myself out loud, I said, I'm going to steal this book. Just that fast. It was just fascinating how it was written. And I walked out of there holding it above the little, magnetic detector. I could have checked it out, right? I was a student at that school. And it's the only time I've ever really done something bad, certainly stolen anything, like that. And I don't know what I did with it, but I just was totally enthralled instantly by it. And I became a molecular biologist right then. I switched all my classes up to like more chemistry, yeah, like that, And then and that was a long time ago. And I only realized like 3 years ago in teaching this class, what was happening there. And what was happening there is literally this. So I loved being out in nature as a kid, and I also liked working on farms and working on farm machinery. So the equipment and the mechanical, physical, matter-of-fact, common sense nature of farming. And that's what molecular biology is. A molecular biologist is literally a car mechanic of life. It's nuts and bolts. It's just nuts and bolts. It's all physical. A lot of it's too small for us to see, even with a strong microscope. But yet we can prove it's this way. Like manipulating those genes is just like that, right? And so that's important to appreciate. You can learn all the stuff in our course because it's just matter-of-fact stuff. It makes sense. The challenge is it's new and there's a bunch of it. But it's just so start today with the confidence of, yeah, you could get all this stuff figured out. But, I don't know much about car mechanics really, like that. I have to rely on Stevie and John over there to, fix my car, and we all do, right? But I'll say to my students, if their car doesn't start, no one gets out and checks the tires, right? Tires are required for a car to go, but not start. And everything in this course is like that, whether it's the molecular biology or the cell biology type topics. It's just all like that. When a person gets cancer, it's all down at that molecular level. And it's a tough, tough story because of the physical changes that are happening down at that molecular level. Yep? Yeah. And so I love that. I'm just freaked out. When I came to USM, I wasn't hired to teach this course. There was another professor teaching it. And I only started teaching this course about 7 years ago. I was here to teach intro bio and developmental biology, like that. And that other professor left kind of abruptly, left USM, and I was just told, like, over a holiday break, Dave, you got to teach molecular and cellular biology in the spring. And this is relevant because this is what happened. I was like, okay, I know how to teach that course. Oh my God. You have no idea how much I love this, right? I'll just start crying in class. Another semester I yelled out the window so loud the other instructors ran down the hall. I was so embarrassed, because it was like if you hear someone shouting really loud in a classroom building, it's like a trigger warning where you're like, oh my God. But yeah, no, totally. I mean, I could do it right now and just shout about molecular biology out to the world. Yeah, super exciting. So here's the deal. The first time I taught this course, I was on break and I was just like, I'll use the textbook that the previous professor had used, whatever. And I got the textbook, and there were 8 authors, and I knew 7 of them really well. They're molecular biologists, molecular and cell biologists. I know them, and I'm old. I knew all these people really well. I know the author of our book, And I started looking at the book, and it was molecular biology writing like a vet book. where I'm like, I'm going to steal this book. I just want to open my head and put it in my head, you know? And so when I came in, the first time I taught it here, not that long ago, and this is what's relevant, I came in that semester and I said, this book is great. It's way too much information, and you won't understand it at first reading because it's, complicated car mechanics. But I'm not going to explain anything except when you ask questions. And that's all we did that semester. And there were only 2 students that really got the hang of it and really asked questions. And I would explain it. They'd say, look, so I'm looking at the book and I don't, page 358. So in this course, I don't do that anymore. I switched books. This one is less like that, written less like that, so I'm comfortable reading it and talking about it, But I bring it to class because you should bring questions to class. Keeping in mind, the first time I taught this, I was just like, and I wasn't being mean, I was just like, the way it's written, and this book is really, really nicely written, it's great. But we jump into it in a way that, it helps me want to explain it more, like that. But there's still a lot of that. So I've already emphasized, when you're looking at stuff outside of class, just write down questions. And you can email them to me, but bring them to class, ask them in class, like that. And that's great. I don't do a very good job pausing, but you should just ask questions anytime. If you have a question, it's really distracting, right? Because it's embarrassing to raise your hand and stuff, So you got to kind of just get that out of your system before you can focus again. So you can ask questions. You can bring in your book and ask questions from any place in the book. Way ahead of where we are, way behind, some other class, anything like that. It's my job to fit that, into our class for everybody else. Yeah? Okay, so what's this going to be course is going to be like? The topics we're going to cover have been so overwhelming to the table that is biology that at almost every college and university, the first semester of introductory biology for science majors is this course of these topics. That's what that course is. It's introductory cell and molecular biology, right? It's some chemistry, it's some genetics, it's some cell cycle, some cell signaling, some metabolism, some photosynthesis, those topics. Those are all of them. That's how much it's overwhelmed all of biology. It's just these topics, the first semester, it's just that. Okay? So we use the Campbell textbook. So here are the chapters that are typically covered in the first semester of biology here, and if you are a transfer student, it'd be the ones you had in your school. And so here are the topics. You don't have to try to read that print. Here are the topics that get covered. And in green are the topics that we're going to emphasize in our course because you already had some stuff on metabolism, And so we're skipping a couple of chapters in our book with intention. We could crank down a pathway of metabolism. But my understanding is that a bunch of you students will have taken biochemistry, and there's quite a bit of metabolism when Pete Woodruff teaches biochemistry, like that. So, there's some stuff we won't emphasize. You have a year of intro chem as a prerequisite for this course. So we're going to start out, that you know quite a bit of chemistry. So we'll do those kind of topics. And in cancer biology, these are 2 biggies, and this is chapter 16 and 17 in our book. So chapter 16 in our book is cell signaling. And, in these intro bio chapters, there are different people that teach the course, and they emphasize different chapters. So you might have said, oh, we didn't have a chapter on cell signaling in my, intro bio course. That's fine. I'm just showing you that that's what that course is. And so you have a lot of background knowledge. The thing is, with that intro bio course, those topics are covered really fast. And so it doesn't really sink in very well. Yeah? Yeah, and so there, see, cell signaling, cell cycle, those are the topics that we're going to be roaring toward, and because it's a bunch of cancer biology. I put this up here. I mentioned already that if you happen to have that 8th edition, the chapter numbering is a little bit different. So back to this, I think cell signaling was the 16, 17, 18 used to be 17, 18, 19, 20. So if you happen to have the 8th, you just have to scramble on that. But again, I don't recommend the 8th because it is different. We won't do this today, but something in your syllabus that's described in the video is normally we stop about an hour into class and we have a very quick concept chat. And I do that in all my classes because it helps you remember and start to recall the stuff you're learning. And when we talk about learning, for me the most important thing is to realize that it's all about recall. If you're just listening to something, like let's say you get some help on directions to get to the mall, right? So you stop your car and you say, you know how to get to the mall? And somebody says that, right? You close your window, drive on, and the first thing you do is you start repeating it. Try to hold on to that learning, right? Same thing with a song. You want to learn a song? Play it, sing it. Oh, I got the first verse. Okay, play it again. Oh, I got part of the second verse, right? And then the more you sing it, the more you know. So we do this in class some to try to start that process of trying to remember what you just learned. You turn the page and try to take notes on what you already read without looking back at the page. What did you get out of that? And practicing that helps. Practicing and getting more familiar with the idea of recalling things as you're experiencing them is good. So we'll talk about that. Then here's some stuff about what this, so we won't do that concept chat today, and we also aren't taking attendance today because we're just getting the ball rolling. This is a hard course. It's just a bunch of stuff. It's a bunch of new stuff. It's a bunch of nuts and bolts stuff. And it's a hard course like I think car mechanics course must be hard, right? There are a lot of different kinds of cars, right? Like a hybrid car. Imagine trying to fix a hybrid car. Part electronic, part gas powered. It's like that. But, you could be a car mechanic, you need to figure it out, right? And, it's really up to the instructor, the teacher, or the car mechanics to kind of take it in a way that you're like, okay, I'm getting that. But, again, have the confidence of, yeah, you can figure it out. It's just a bunch of stuff, okay? And it's like this. It's all physical. It's just super, super physical. And it's just a bunch of parts, and the parts are interacting. And they're all down at that cellular and molecular level. And we zoom in on them, like that. And we'll do assignments where we draw it out, try to draw it out, label out, to try to get a handle of physically, right? A bike's pretty simple, and still, you start zooming in on it, and you're like, I gotta take a couple links out of the chain, I gotta do that. It's not been done, I haven't done it yet, because I don't know how to do it. So I'm gonna learn about doing it, and I'm gonna do it. Oh, and I already described the first time I taught this course. Then, here are the best ways to learn for this course. So get active. It's physical. You can draw everything in this course out. Draw it out big, label it. We'll have assignments to do that, but you can just do that. Take notes when you're reading the course, take notes in class, use a class recordings to go over them within a week. Write down questions, bring them to class. Whether you're looking at the book, you're looking at the video, you're in another class, right? Oh man, I love that. Here's the exam format. We're gonna have 3 exams, and they're all the same format. They're going to be 50 short answer questions. Each is 2 points. Sometimes they get kind of combined together, like it'll be a question that's worth 6 points because it's several things. But it's like that. It's like, what's this? What's this? What's this? It's flashcard type knowledge. Like what's the FOXO protein do? And you flip it over and look at it. Something I realized a few years ago that works, I think, great is to make some flashcards, but actually make them based on labeling a figure, and then tape some big pieces of paper together and make a big drawing so that you're actually labeling it using your flashcards. And I think I have an example of that. Yeah. Here's a good example. It's from chapter 16, but we're going to get there really soon. And this shows a cell, and it shows this response of FOXO, a protein that's found in us and in every multicellular animal. I think that's pretty safe to say. So, not just humans. And it shows how this protein inside the cell is responding to a molecule, like a hormone, outside the cell. And so there's the cell membrane, and this just shows there's a receptor, the factor binds the receptor, and in the absence of the factor, this FOXO protein is allowed to go into the nucleus and it do its job, and its job is to activate gene expression. In the presence of the growth factor, there is a signal transduction cascade. The FOXO protein gets phosphorylated, and another protein, 1433, binds to it and sequesters it in the cytoplasm. This FOXO protein needs to physically be in the nucleus to do its job, to regulate gene expression, because that's where the genes, the DNA is. And it can't do its job in the presence of the growth factor. So that would be a good one to draw out big. And that's cell signaling. That's the focus of that chapter. And the purpose of that chapter is to explain how we can regulate transcription of genes with signals coming between cells. We won't spend hardly any time in this course talking about the significance of these little nuts and bolts things, right? Like that. You know, like to say, well, when you get the lug nuts on the wheels, you can drive yourself down to Florida. That'd be fun. And here's some postcards of what Florida looks like. We do almost none of that, because a molecular biologist is literally a car mechanic and has zero interest driving cars. They just get such a kick out of just looking at how the nuts and bolts fit together and how the little carburetor works. Yeah? Here's just an example. So this FOXO protein I mentioned is found in every animal. And about 30 years ago, a scientist named Cynthia Kenyon started studying this little worm, a little nematode worm that you might have heard of called C. elegans, Cyanorhabditis elegans. It's just a soil worm. It's almost microscopic. You can just barely see the adult with your eyes. And you can grow it in lab, and scientists were studying it at lab the same way they study laboratory mice or fruit flies and so forth, like that, yeah? So there's a little nematode, and Cynthia's working on it, and it just happened that the life cycle of this worm, the natural life cycle out in nature, had a real curious feature to it. Normally, if there were lots of nutrients, the little worm would grow up fast and reproduce and die in just a couple of weeks. It had a very short life cycle. However, if there were challenging conditions in the environment, for example, nutrients were limited, the temperature was uncomfortably hot, or maybe most interestingly, it was crowded with other worms of the same species, the nematode had an option of, instead of growing up, it could go into a kind of hibernating state. And it made a kind of a thick skin to prevent desiccation, but it kept these little chemosensors out detecting the nutrient availability and also a pheromone molecule that all the animals produce. Because when it was crowded, that pheromone scent was there, and it would stay in hibernation. And it could stay in that hibernating state, it's called a dower larva, for 5 months and come out and reproduce and die. And what this Cynthia Kenyon did is she worked on this in a lab, and because of that special feature of that animal's life cycle, she discovered that the animal has to actually slow down its rate of aging when it enters that hibernating state. You might think, nope, it's just hanging out. It actually actively slows down its rate of aging and extends its lifespan from 2 weeks to 5 months, so tenfold. Human lifespan maybe 80 years, 800 years. That kind of scaling difference was being done. And she was a geneticist, and she studied it by identifying genes that were mutated and either failed to do that or could start allowing an unhibernating worm to age more slowly, extending that lifespan without making the environment harsh. And one of the genes for this protein she discovered is FOXO. When FOXO goes into the nucleus in the absence of this growth factor, it activates hundreds of genes that are involved in slowing down the rate of aging. We have this protein in us. In the human population, there are different alleles of this gene, just like there are for any gene. And there are some families that famously live to be really old, and they have a certain allele of that gene, right? So the stories of the proteins that we're going to study are just crazy, because it's all about everything that's interesting in our life and nature. Yeah, but we'll just be like, yeah, FOXO, kind of a cute name. It's an acronym for a bunch of dumb words like FORCAD and stuff like that, but it's got a cool job. For us, it's just, look, it is so sweet. It's got to be in the nucleus to do its job. Who cares what its job is? And it can't go in there if it gets phosphorylated. Wow, right? Like that. Like get a tattoo, like with one arm. Oh, look, FOXO is excluded from the nucleus. Oh, look, FOXO's in the nucleus, right? Because it's physical like that. All right, and so that's what our exams are going to be like. You know, what about FOXO? Or give a list of transcription factors, you know, and explain how are they regulated. We're going to make a list of a dozen transcription factors and how they get turned on and off, like that. So we're doing great. Oh yeah, because we have till 10:45. Yeah, we're great. It's been a long break. Oh man. So, getting ready for the exam. The first exam we'll have is on the syllabus. I haven't put too much of the schedule on the syllabus because we'll see how we do, right? We're definitely going to handle the chapters in that order, but how fast? I don't know. You know, bring in a bunch of questions. When I was in college, I had so many questions that twice my classmates booed me. They're like, boo! Because I was intentionally stalling. The more questions you ask, the less you have to learn. You know? And I've never seen that at USM. First of all, I've never seen students booing, but I was just like, forget it, man, I'm slowing this class down. I'm like, you know, excuse me, professor, but did you have any pets as a child? You know, come on. And people are like, boo, we want to learn. And I'll be like, yeah, take it easy. Yep. So some slides about that, about taking notes and getting physical. It's a very physical thing, and you can't just, oh, I'm thinking about this. I've got this in my head. It's too many parts, right? That's what this video is all about. Make a clay model video. We'll have some assignments where we do that. Break it down like that. We'll have to draw a gene on an exam. It's really easy to talk about a gene. We'll have to draw it. We'll have to draw this out and explain it on an exam. And it's fascinating. So, and doing that, and maybe this will be our last slide. No, it's not, because we have another while to go. I keep thinking we're ending at 10:30. We're doing great. Here's the first good pausing point so far in our course. We have our first big picture. And our first big picture is that in the muscle videos you're going to watch, you're going to see, well, the cells have the muscle genes. They're just not using them yet. So they must use them. How do they use them? How does it get started? And the answer is the DNA doesn't change. Proteins that bind to the DNA change. And those proteins are called transcription factor. FOXO is a transcription factor. And it's going to change the gene expression. So they're the dynamic story. The muscle story is dynamic. Here's some cells. They're not muscles yet. No, they're muscles. How'd that happen? And it's very well understood. Nobody snapped their fingers. There was no lightning bolt involved. We activated a set of transcription factors. And so, like I said, we're going to make a list. And our first one on our list is going to be one of the transcription factor proteins in the muscle videos. Myogenic. That'll be the first one on our list. How does it get activated? How does that switch work? And by the first exam, we'll have a list of about a dozen. FOXO will be one of them. How does it get regulated? It gets phosphorylated. It gets turned off by phosphorylation. Yep. Okay. And so just more stuff about FOXO, I guess, a little bit out of place, but FOXO, when it gets activated, it binds to DNA and it turns on a bunch of genes that are involved in a bunch of things like that. Here's a figure from a different textbook, and it shows that original worm, nematode worm. They didn't call it FOXO, everybody calls it FOXO now, DAF16, and they found the same research in mice and flies that when they manipulate that gene and organisms, they can increase cellular lifespan like that. So just fantastically significant story. There's Cynthia Kenyon. I guess I got those slides a little bit out of place. Here's a little bit that's kind of funny about Cynthia Kenyon is, right, so she's now old, right? She was your age in the 1970s, 80s, like that, right? She's pretty old. She was a professor at the University of California, San Francisco. And you haven't heard of that because it's just medical buildings. The campus is not an undergraduate campus, it's just medical buildings in downtown San Francisco, or in San Francisco. And she was a professor there for a long time, and she's a pioneer in the study of aging, okay? Just doing her thing, you know? And you know, Google is a big company with a lot of money. And a few years ago, Google did this kind of financial rearrangement where Google got bought by a company that Google invented called Alphabet. So now Alphabet owns Google just because Google created Alphabet to own it. And they're doing that hoping that nobody noticed that then Alphabet had all the money. And Alphabet started some companies that are super secret. And one of them is a company called Calico. And Calico just has billions and billions of Google dollars they're spending. And they're spending it secretly, and they hired Cynthia Kenyon. Because Calico stands for the California Life Company. And they're just studying aging and try to slow down the rate of aging, you know, medical products. Because they're like, oh, you could teach these students about cancer. Well, cancer is mostly a disease of old age. So why don't we just slow down aging and start to eliminate issues like cancer and heart disease by just having people be younger and have more active immune systems like that. So yeah, she just went dark, right? Cynthia Kenyon used to be very at all the meetings and very talkative, and then she's just gone like that. Yeah. It's fascinating to see what they do because you hear little hints of it on the news. Like a couple years ago, there was a black barge that got pulled up outside of Portland, like out in the bay a ways, and people were like, "What's that?" And some people were like, "That might be something that Alphabet company's up to." And then it just got pulled away after a while. Yep. Yeah. It's fascinating. Why would they show up in Maine? They have everybody knows they have a collaboration with Jacqueline Lab up at Bar Harbor. And the collaboration is just interesting to hear about. So they've been able to manipulate genes in mice really well. And they can use that as a model system for these kind of studies. And again, we're not going to tell stories like that in this course, even though you're like, well, that was the best part of this class today. Too bad, you know, you're like, I really am not into nuts and bolts like Dave is, but they're cool. Like you just twist them on and they stay put. Keep the tires on your car. So mice grow old really fast. Mice only live for a couple years. They're rodents, and in general, rodents are small. They have a high metabolism. They don't live very long. There's a rodent that lives a long time, and it's a naked mole rat. And you might have heard of naked mole rats because they have them in science museums for kids and stuff like that. They live underground, they're blind, blah, blah, blah. They have a lot of curious features, and one of the curious features is naked mole rats live for almost 30 years. They're a rodent that's really closely related to mice, but they live 10 times longer. So they have FOXO, they have all the same genes as mice, but their rate of aging is slower. Aging and the rate of aging is a selectable trait. It's an evolvable trait. The reason that mice live fast and die young is because they're fitting into a specific ecological niche that they evolved through natural selection to fit into. And just like their fur and so forth, just like their physical features, rate of aging is a phenotypic trait. And naked mole rats just evolved to age slower. So they have the comparable genes, but there are some of them that have been changed. And so everybody knows that Calico and Cynthia Kenyon are working with Jackson Labs in Bar Harbor to bring those two organisms together in research and say, what's the difference between these closely related rodents that lead one to live naturally, normally, 10 times longer than the other? Yep.
Do they have slower heart rates?
Do they have slower heart rates? Yeah, I don't know. There's a bunch of, I love that.
I just heard a theory that all mammals have about the same amount of heart beats.
Yeah, that's a really interesting point.
Yeah,
I mentioned, mice are small, they have a high metabolism. So animals that are small and tend to have a high metabolism, there's a correlation. You can make a graph like that, right? Big whales, big elephants, they live a long time. So there are some curious features that folks have known about aging for a long time that are species specific, yeah? And now scientists are down at this molecular nuts and bolts level saying, oh, their rate of aging is different, their size, their metabolism is different, but they have the same genes that are regulating the process. So let's study those. Yeah, different genes. Yeah, super cool stuff. From this, I think we're doing great. And here's the first example. And again, you haven't watched the video, but you will because we'll be getting ready for it. And one of the take-home messages from the videos that will be the basis for our quiz on Thursday, so the second set of videos, one of the take-home things from that is the videos explain a universal example of gene expression. So I picked muscle and muscle differentiation because you know that. You've had courses in physiology, you know muscles are what cause our arms and legs to move, and they work by having proteins like myosin and actin slide across each other in the striated muscle fiber. So you know about the physiology of them. Well, those are all proteins that have to get made. So how to get made? Well, there's a gene for every protein. So there's a gene for FOXO, there's a gene for myosin, there's a gene for actin and troponin T and titin and so forth. All those muscle proteins that get made, they have a gene that they use to make the protein. So how does that get activated? And the take-home message is, well, let's look at an example gene, and we'll look at the example gene myogenin, and we'll see that there's a transcription factor called MyoD that activates it. And the reason that MyoD does its job is because MyoD is a protein that also needs to get made. So one way to regulate gene expression is just to regulate whether or not the transcription factor gets made. And if it does, it turns it on. So that's our first example. And that's just, again, you haven't seen the videos, it's the first day of the semester stuff, but you will. And that's kind of one of the take-home messages. We got to understand everything that's going on in the videos because there are a lot of different pieces that are at that molecular level. And there's a major take-home message, and the take-home message is going to be, there's a gene, it's got these parts, and it's regulated in this way. And we could make let's see if I have that screenshot. So these are two cells, one that's a little bit younger, a little bit older. This one is a muscle precursor called a myoblast. That one's a muscle. What's changed is the cell became a muscle. It's the same cell, just younger and older. So two cells side by side, and we're describing what's changed in them, okay? So that's a universal example. We often want to see two cells and what's changed, yeah? And the focus is changing gene expression. So we could make the same drawing and just change the labels. And here's what we would change. What are the cells we're talking about? Are we talking about a muscle or a brain cell, a cancer cell that got exposed to a medicine, before and after? Two cells. What are the two cells we're talking about? So that's one label we would change. Then another change is, what gene are we looking at? In this case, it's myogenin. That would change. The pancreas. Well, there are precursor cells to the pancreas. They're not making insulin. There's pancreas cells. They're making insulin. Let's look at the insulin gene. So change the cells we're looking at, change the gene we're looking at, and change the transcription factor. This is MyoD. There are different transcription factors that activate expression of insulin in the pancreas. So we already want to sort of foreshadow that we're using this example because it's a universal example, not limited to humans or eukaryotes. It's true for bacteria as well. Unfortunately, the terminology for the nuts and bolts naming varies in some different settings. So when you take microbiology, they don't really say, oh, this gene is just like that myogenin gene. There's features of it, and the key significant features, they're always the same story. And remember that molecular biology emerged as a field, bringing biochemistry and genetics together to explain this. When people say, hey, what's your definition of a gene? Right? That's the basis of genetics, right? Gene ticks, right? Most biologists are like, whoa, it's really rugged to define a gene. That's hard work. And a molecular biologist is like, what? You know, like a car mechanic is like, it's hard to talk about carburetors. And, a car mechanic's just like, what? Yeah, because the first thing you'd say is, well, it's physical. When you talk about a gene, it's a physical thing. And that's one reason why most biologists still think it's kind of hard to talk about. Because the field of genetics is just a little over 100 years old. And for the first half of the history of genetics, nobody knew what a gene was. It was theoretical, and it's not now. In intro bio, when you look at the text, the students don't appreciate it, but they're picking examples to try to convince you of that. Even today, that the gene is physical, right? They pick simple examples in genetics. Look, you cross the red allele and the white allele, and you're going to get a pink allele. What they're doing there is they're still trying to convince people this is a physical story. And part of it is genetics out in the real world is always way more kind of seemingly subtle and complicated than that. But part of it is just to say, yeah, no, that's the real deal, right? And so it's amazing because when we talk about a gene, we got to start with this video. You know, in principle, you've all had genetics. A couple of students didn't, but they've had other courses that fit, you know. But yet, we still have to start out with it. We're trying to work toward a definition of a gene, and then gene regulation, and then transcription regulation. You know, we already looked at FOXO, foreshadowing chapter 17. Chapter 16 is mostly about switching transcription factors on and off, you know, to get that change, to have the cell, oh, now I'm going to make a pancreas. Yep, super cool. Yep. So, first example on that list, and again, by the end of by the first example, we'll have about a dozen on those, and those are just hallmark, famous, cool examples of how to make that switch occur. Okay, so we're doing great, and so we can add our second one, because even though we're not really looking at that chapter in our book, we've seen it in class. Oh, we're going to talk about a transcription factor named FOXO, and FOXO gets regulated by phosphorylation. And when it's phosphorylated, it's off because it's excluded from the nucleus by grabbed by 1433. So our first example is going to be myogenin regulated by MyoD. And I guess I just failed to get MyoD up there, but I'll put that up. It's a transcription factor. And it works by just getting made. If it gets made, we're going to make muscles. It's really natural after watching these videos for a student to ask, and it's a good question to ask, well, how does MyoD get activated? And the answer is, it gets complicated. Some stories in the gene regulation become complicated stories, and it's complicated. What we're actually going to get out of this is a little series of steps where we kind of jump into the middle. So I always say, like, imagine dominoes falling. You've seen that, right? People stand up dominoes, and they knock them down. And what we're doing is we're jumping into the middle, and we're just looking at 3 dominoes that are falling. And we don't worry about the upstream dominoes, just these 3. And the first one is MyoD. MyoD is a protein. There must be a MyoD gene, and when the gene gets activated, the domino hits this domino, the MyoD domino falls. It falls by making MyoD. The MyoD protein goes into the nucleus and does its job. it knocks down the next domino. The next domino is a myogenin gene. When MyoD binds to the myogenin gene, the myogenin gene gets activated. It turns out that the myogenin gene is also a transcription factor, and it also works as soon as it gets made. So as soon as myogenin gets made, the myogenin protein binds to target genes that are muscle-specific proteins like actin and myosin. And there's going to be a bunch of little dominoes that fall next, but the next dominoes to fall are the muscle genes. So we're just grabbing a little short snapshot out of a series of dominoes falling, about a series of gene expression changes. And the one that's at the center of our little story in the path is myogenin, because it's such a sweet example. All you need to get it going is MyoD. And all you need to do is make MyoD, and then it's going to go. And then it gets made, and all that myogenin does, its only job, is to bind to muscle genes and activate them. And there's some other proteins that help, but that's the deal. Yep. So now we do have to stop. Yep, we're done. All right. And again,